Can you guys hear me okay in the back? Okay, just let me know if it fades away. I'm excited to talk to you about the vaginal environment as a modifier of HIV risk. Now, I'm going to talk about the optimal vaginal microenvironment, and you might note I'm going to try to avoid the word abnormal. Abnormal is a little bit challenging to define. What's abnormal? Are you abnormal because you have symptoms? Maybe. But what if those symptoms are a little bit difficult to call something abnormal when the prevalence is 60% in some sub-Saharan African settings? And if you go to many sub-Saharan African countries and you just looked for bacterial vaginosis, which is what we're going to talk about, you'd probably find it in about 60% of women. So I'm going to try to avoid that term. I will slip into it, I'm sure, but just FYI. So I'm going to talk about an optimal vaginal environment, and how do I define that? What's optimal? Well, you would like to think about an optimal vaginal environment from the standpoint of what outcomes you would like it to support. What's the point of having a vagina from an evolutionary standpoint? It's so you can have babies, right? And you can perpetuate the species. So you want a birth canal that is going to give you the highest likelihood of having healthy babies who are uninfected, who are normal birth weight, and who are full term. Okay, so that to me is really from an evolutionary standpoint and a reproductive health standpoint the optimal outcome. But there are other outcomes too that we'll talk about, and they might relate to being symptom-free, right? They might relate to being sexually healthy, whatever that is to whoever you're talking to. The other way to think about it is, okay, you've defined these outcomes, but what are the microbiologic and immunologic parameters that predict those outcomes, okay? So how do we know if we look at a vagina in a point of time that it is in an optimal state to support those kinds of outcomes? So we'll talk a little bit about those too. We'll talk about implications, particularly with regard to HIV. Given the brief time we have together, I can't possibly talk about all of the implications. For example, there's a lot of really interesting work, mostly in the laboratory, looking at the effect of vaginal lactobacillus on chlamydia trachomatis, but we can talk about that at the break if we have time, but I'm really going to focus, given our emphasis, on HIV. And then I'll talk a little bit about what's next, okay? How could we possibly find what we want to come out of it, literally and figuratively, could support a healthy vaginal environment. And then now that I live in the South, I can show slides like this right now. So one-stop shopping, guns, wedding gowns, and cold beer. The idea of combination prevention is you have a one-stop shopping situation where you can get a ring that maybe prevents acquisition of HIV, prevents acquisition of herpes, and maybe even prevents unintended pregnancy. So that would be the goal, right? You would really like to have a package where you could try to give women the optimal chance to protect themselves from things that they shouldn't or don't want to experience. So this is kind of how I've put it together in terms of the outcomes domain. And I think for whatever you do in clinic, I think even if you're a clinician who never, or if you're a person who's never looked under the microscope or done a pelvic exam, I think you can probably appreciate this. So the reason that bacterial vaginosis, which I'll define shortly, or abnormal or suboptimal vaginal environment really got on the map in the late 80s, sort of the 1990s, many of you in this room were involved in this, particularly the STD clinic clinicians, it's because it became pretty clear that not having this optimal vaginal environment was associated with adverse reproductive health outcomes, right? So you were more likely to have a healthy baby, meaning a normal birth weight baby, a normal term baby, and an uninfected baby if you had an optimal vaginal environment. When you had BV or bacterial vaginosis, the risk of early delivery, preterm delivery, neonatal infection, and low birth weight all went up pretty significantly. Women who had a suboptimal environment, i.e. BV, also were more likely to have chorioamnionitis, which is an infection of the chorioamniotic sac, so an infection of pregnancy that's also associated with premature rupture of membranes and preterm delivery. So clearly there was something going on there. People got very interested in that. And that's remained a huge focus of BV research. The other big area, of course, is that people realize that clinically these sort of abnormalities or suboptimal situations were associated with vaginal symptoms. So bacterial vaginosis, or BV, the most common cause of symptomatic vaginal evaluation, or that's the reason that women go for these types of symptoms to their providers most frequently, causes abnormal discharge and abnormal odor, probably about half the time when you got it. We also found out that bacterial vaginosis was associated with cervical inflammation, and that many of the bacteria that we saw with BV could also ascend to the upper reproductive tract and cause PID. And indeed, that's one of the reasons that we include metronidazole in our PID regimen, or in many PID regimens, because you really would like to cover the anaerobes associated with BV, and I'll come back to that in a minute. I think the most recent, I would say last 10 years or so, have more focused on the aspects of BV, or the vaginal environment, related to sexual health, right? So certainly sexual health relates to not having symptoms, right, and not having abnormal odor, not having abnormal vaginal discharge. But it also relates to having an optimal protection from getting super infected with STIs and HIV. And that's what people have really gotten interested in the last 10 years. It's become very clear that to have this optimal vaginal environment is a very important first defense mechanism from getting HIV in particular, and from getting other STIs. So we'll talk about that in a little bit. Now, this talk can never be all things to all people. So this slide is a bit of a way to inoculate myself from criticism from people who say, well, why didn't you talk about this? Why didn't you talk about this? So this, if you really want to geek out on this topic, you can take a look at this review article that Caroline Mitchell and I wrote. And I really like this graphic. I'm sorry, it's a little bit blurry. But what it's saying is that if you go down to that dark cell on the left-hand lower side, what it says is that the thing that mediates probably all of the bad stuff around having a suboptimal bacterial community or inflammation in the environment is mucosal inflammation, and that does increase your risk of HIV, STI. But what gets you there? How is that generated? And it's really complicated. You could spend an entire half day on each of these areas to talk about the subtleties of, for example, how do hormones affect the vaginal environment? Clearly, hugely, right? They don't only just affect the hormonal environment, but they affect the microbial community, which in itself we know is mediating some of this stuff. There are probably host factors. Erin De La Cruz, who works with David Fredericks, has a couple of beautiful papers coming out from our data and our participants here in the Vaginal Health Clinic that showed that host genotypes, namely around TLR polymorphisms, impact whether you're likely to be colonized with certain types of vaginal bacteria and whether you're likely to generate inflammation or inflammatory mediators with the presence of those. So that's very complicated. Microbial metabolites. Obviously, bacteria do not just hang around and look pretty on the stains that you make. There's a lot going on. And some of these metabolites, like short-chain fatty acids, actually are critical immune mediators. We know this from a lot of work in the gut. Lots of a focus on these short-chain fatty acids in the gut, but they are also in other body places like the vagina, and that probably plays a big role. And then, of course, we can't forget sexual partners, right? What's the main mediator of HIV risk? It's your sexual partner's viral load. There's no getting away from that, right? You can have the perfect vaginal environment, and if you're exposed to a partner who has 1 million+ copies of HIV and semen, it's going to be very tough to overcome that challenge. So not only that, but sexual partners, we know, transmit bacteria. So BV-associated bacteria, I don't have time to get into this today, but very clearly are prominent causes of urethritis in men, particularly men who are uncircumcised. So these anaerobes very much like the foreskin, like to live under there, and that's the reason that circumcision is almost certainly a risk factor for bacterial vaginosis, and probably one of the reasons that, I'm sorry, not circumcision, being uncircumcised, probably one of the reasons that being uncircumcised is a risk factor for HIV transmission to women. So just to give you a sense of how rich this field is, how much it's evolved, and for those of you who are looking for something to work on, there is no end of interesting things to take a look at. Okay, so let's try to look a little bit at the vaginal microbiome, and I think I've got a pointer on here, yeah. So just remember, how many of you in the room have done a gram stain of vaginal fluid? Sylvan, wow, that is amazing. Let me just tell you, there are not many places in the world where you would give a talk and you would go and you have people say that. So that's very exciting. So Bob Nugent, some of you don't know that Bob Nugent is a real person. He's actually married to Heather Watts, who some of you may remember, who's a very important OBGYN, infectious disease person. Bob and others generated this score called the Nugent score back in the 80s, and what the Nugent score did was simply to gram stain vaginal bacteria and to quantify on a scale the number of morphotypes that reflected lactobacillus. So those are the long purple gram positive rods there, contrasted to other morphotypes that represented a spectrum of anaerobic bacteria. They were most interested in Gardnerella morphotypes, in Mobiluncus morphotypes, but things that really, as you can see when you shift from that really beautiful vaginal environment to the left to frank bacterial vaginosis on the right, really gives you a sense of how much things are changing. So that was the Nugent score from 0 to 10. It was the original sort of research standard that we used to define bacterial vaginosis. And it gives you a visual sense, right, of how you go from this very monotypic environment, which is really unusual. It's exactly the opposite of the gut. We know that diversity in the gut is a really good thing. When you go to a more sort of dominated environment in the gut by a certain kind of Bacteroides or other species, it's not so good. In the vagina, you really want to be dominated by those lactobacillus species. So that was our first sort of sense of that spectrum. And what we've done now has really been to shift to molecular analyses of the vaginal microbiome. What's interesting, though, is that it pretty much is telling us the same thing. So I'm going to give you a couple of other examples, but down the bottom there, what you're seeing is the very similar spectrum using 16S ribosomal RNA sequencing, ribosomal DNA sequencing, showing you on the left-hand side 2 community types that are lactobacillus predominant. This is a study that I'll talk about a little bit more later. Going all the way to the right where you've got incredible diversity, all of those different colors are different species, and it's really showing you the same thing. So that's kind of what our descriptive understanding, I would say, of the vaginal environment is. One key point about the normal situation that's really important, it's dominated by lactic acid, which, as it turns out, is probably the main mediator of protection, interestingly enough. And it's dominated by a low pH, less than 4.7, because of that lactic acid. Okay, so low species diversity, low Nugent score, low pH, and lots of lactobacilli with lactic acid, and that's really what we're talking about. Now, a couple of years ago, so you might say, okay, well, that's great. So there's lots of anaerobes. Is that all it is? Is it just a syndrome of bad odor, anaerobes, lots of discharge, or is there something going on there? Is that disrupted community down there really just one thing, or are there different types of BV? And that's been a question, I would say, that we've been interested in for a while. This is a study that we did led by Sujatha Srinivasan in David Frederick's lab, published a few years ago, that looked at women who were attending our STD clinic here in Seattle. And I like this because essentially you're now looking at this sort of analysis flipped on its side, okay? So, and you're going from the most lactobacillus predominant species down the bottom there, in the red and the orange, and you can see those green bars represent the Amsel and Nugent scores or Nugent criteria, and basically they're saying that these are women who don't have bacterial vaginosis by those typical criteria. And Amsel, I should mention, is the clinical criteria, which we can talk about in a minute. So those were very quote-unquote normal women, but you can see that there are 2 major populations here, right? One of them is dominated entirely by Lactobacillus crispatus, which we have known for a long time is sort of the most protective or most associated with positive outcomes in the vagina. But then you've got this other group down here in orange that's dominated by something called Lactobacillus inners, which was kind of a surprise, yet we still have a normal pH and we still have, by and large, pretty normal clinical presentation. So that was kind of curious. And then you go up here and you start looking at the women who have clinical bacterial vaginosis, and you can see that there are clearly subgroups of women. So here's a group of women, and I should mention that each of these lines is a single woman, and as you're following across, you're seeing what their Amsel score, the Nugent score was, and then what their bacterial relative abundance is in the vagina. What is this? This green thing is something called Smithia leptotrichia. Why is that interesting? And you can see there are women down here who are dominated by that too. Smithia and leptotrichia are a species that has been very consistently associated with urethritis in men. So a big cause probably of urethritis, and it's also one that we have found to be most inflammatory in the vaginal bacteria, associated also with endometritis and upper reproductive tract infections. So I would wager that these women are probably women who are getting their BV, maybe maintaining their BV by continued re-exposure to their male partners. Okay, other women in this group, probably not so much. They have very different compositions. So what we learned from this study is that women with BV have diverse heterogeneous communities, and those who don't have it are pretty much dominated by either of these lactobacillus species. Okay, so taking that a little bit further, again, thinking about the fact that BV is really common. If you go to our STD clinic here and you just look for it, you'll probably find it about 26% of the time, so 1 in 4 women who come into the clinic. If you go to any of the clinics in sub-Saharan Africa where we enroll women for HIV prevention studies, you probably find it between 48 and 60% of the time. So it's really common. Clearly, lots of women have it but don't have problems. They don't have PID, they don't have abnormal outcomes of pregnancy. So there has to be something going on there where maybe this bacterial diversity plays a role. And it may also be those other factors that I showed you in that really scary slide before that talked about the host, that talked about partners, that talked about all other things. But it's clear that there is something going on with a mix of species. And what Sujatha did here was a very sophisticated analysis to look at the interplay between these species that we found in these women and whether they were associated with individual components of the AMSL criteria. So the AMSL criteria for BV are what we use in the clinic to define it. So what it says is that if you have 3 of the 4 things noted here, you are having a clinical diagnosis of BV, and that relates very well to AMSL criteria. It's abnormal pH, it's positive whiff test, which means it smells fishy when you add potassium hydroxide. It's abnormal vaginal discharge, and it's the presence of clue cells on saline microscopy. And clue cells are just vaginal epithelial cells with a lot of vaginal bacteria adherent to them. And what I want to point out is that in this analysis, leptotrichia, in particular, also Eggerichella, Adipobium, Gardnerella, were the most associated with more AMSL criteria. So they were clearly kind of major players or signal bacteria in this process. And again, coming back to leptotrichia, it's interesting because we know that that stimulates inflammation in men. So just getting this sense that there's probably something a little bit more complicated going on here. And this is going to come back when we talk about HIV acquisition. So I'm going to try to pull this all together, and it may be hard, but we'll see if we get there. So what do we know from all these studies? And there are many, many other studies I could show you. I just like those because it's our local work. I think it's beautifully done, and also because I think it lays out a lot of these principles. So I think all bacterial vaginosis is consistent in having an elevated pH and loss of non-inners lactobacilli, which probably accounts for the consistency of adverse reproductive and sexual health outcomes, right? There's a common pathway. You don't have the right kind of lactobacilli, and you have some consortium of bacterial anaerobes. Otherwise, though, I think there are discrete types of BV that can be defined by distinct communities of these species, and that probably directly impacts your local immune environment. And that's probably an individual pathway that may be related to the host genotype, or it may be related to who you're having sex with and how you are continually exposed to these bacteria. Okay? So let's take a step over to the HIV world and try to relate those observations to some recent data that we have seen with HIV, because it's really gotten very, very interesting. And again, keep in the back of your mind the fact that we still don't have a great way to control HIV in women, right? PrEP probably works, but there are issues with PrEP, and I'm going to talk about those in a minute. Okay. So there's a very nice meta-analysis by Julian Adashili done in now 10 years ago, actually, that took all the older epidemiologic data looking at risk of HIV acquisition in women by traditionally defined bacterial vaginosis, so pretty much Nugent score, some AMSL criteria, but mostly Nugent score. For those of you who remember Hal Martin, that's Hal's study, which was one of the first very nice studies. And the bottom line is that you probably overall, if you look largely in women in sub-Saharan Africa, have a 60% increased risk of getting HIV when you have BV, even if you control for other STIs. Very importantly, these studies, what's the most important STI to control for when you're thinking about Africa and HIV acquisition? Herpes, right? So this controlled for herpes is controlled for acquisition of chlamydia, gonorrhea, and syphilis. All these studies were really very rigorous in doing that. And you still come up with this 60% increase. Now, is an increase of 60% or an odds ratio or hazard ratio of 1.6 a big deal? Well, think about it from the standpoint of population attributable risk, right? For those of you who are epidemiologically inclined. Again, I mentioned 60% of women in sub-Saharan Africa have BV, and when you're looking at places where the HIV incidence is 8%, that's a huge attributable risk for having a suboptimal vaginal environment, okay? So it really does play a role. So that's what we've known. Now, take that old epidemiologic data and let's fast forward 10 years and say, okay, since that time, we've gotten better at characterizing the vaginal microbiome, as I mentioned, by molecular techniques, right? We have gotten better at characterizing the immunology of the genital tract. It used to be that we just looked at cytokines. We still do look at cytokines, but they're really kind of not that helpful because I think of them as little smoke. You know, we really don't know what the fire is and what causes it and what's fueling it, but cytokines are there, and so we see them and we're like, oh, wow, something's inflamed. So we've gotten more sophisticated at that. So I want to talk to you about, I think, a really pivotal study that was published earlier last year by Lisa Gossman and Doug Kwan's lab, and it's called the FRESH study, and I don't know how many of you have heard about it, but I think it's really good food for thought. So what the FRESH study did was to follow young women who are 18 to 24 for HIV acquisition in KwaZulu-Natal. And what they ended up showing, and I'll give you the punchline before I take you through the data, was, again, not terribly surprising, right? We already knew from this old epidemiologic data that it wasn't good to have BV if you were at risk for HIV. They did confirm that, but what they did very critically was to do very careful microbial analyses using these new methods, and then they also did cervical biopsies to look at CD4 cells to see what the presence of these vaginal anaerobes were doing to the receptivity of these cells in terms of HIV. So let me take you through some of what they did. So they followed 236 young women in South Africa, and they followed them for almost a year. It was interesting, this wasn't just an observational study, it's a depressing finding because what they did was to bring them in every week. They gave them very intensive skills training, job training. They were very successful in getting these young women onto success in terms of education, keeping them involved, sort of a lot of self-empowerment, and despite that, the HIV incidence was still 8.4. So again, this was, I think, probably one of the most sobering socio-behavioral studies that I've seen in a long time. So keep that in the back of your mind. Well-funded study. Doug's at Harvard. I mean, it was really, really nicely done. And what you're looking at there which is the first graphic that I showed you on that first slide to give you an example of that vaginal community, are the community types at baseline, okay? So they sequenced something called the V4 region of 16S, ribosomal DNA, and they measured immune mediators, as I mentioned. And what you can see there is you've got these 4 community types, and really it looks very much like that picture I showed you of Sujatha's data from our STD clinic women. So in the orange there, those are the women, community type 1, who had lactobacillus crispus. First thing you know is that there weren't very many of those, right? So these women are starting off not particularly well equipped to resist HIV, at least from the standpoint of what their vaginal microbiome is doing. When does that happen? Probably when women start to have sex, and that's another topic for another time, but keep that in the back of your mind because that's really not very good, right, for 18-year-old women to have so much of the right-hand side. Community type 2, again, coming back to our friend Lactobacillus inners, Lactobacillus inners doesn't make lactic acid. Clearly, you don't have an anaerobe predominance, but look how many women had a community completely composed of that, and I want you to remember that when you see the HIV acquisition results. Then, of course, you get to type 3 and type 4 with increasing complexity of species and increasing diversity, I guess is how I would say it. Okay, so what did they find here? And I'm going to blow up the most relevant parts here so you don't really have to take a look at it. But let me just take you through it. So that is the HIV incidence up in the upper left-hand side, and I already mentioned what the HIV incidence is. And what you're looking at here is, among all participants, what was the prevalence of HIV infection or the incidence of HIV infection by these community state types. And what you can see is that if you had community type 1 down here, you did not get HIV infected at all. And I'll show you that in a minute a little bit better. But these 3 types, very high rates. When they took out chlamydia, which was an independent predictor, not surprisingly, incredibly high rates of chlamydia. That's a topic for another time too. STI is completely neglected in this population. You can see here that those associations remained. What I really want you to look at is the actual data that informed those curves here. So there are your community type groups, type 1. So remember type 1, Lactobacillus crispatus, type 2, Lactobacillus inners, 3 and 4, increasingly diverse anaerobic bacteria. No woman who was Lactobacillus crispatus predominant got HIV, which was pretty striking. In the Lactobacillus inners type, look at the incidence there. Actually, only 66% of them remained HIV negative, so a third of them went on to get HIV. And then, of course, in CT3 and CT4, the rates were the highest. And that's actually a great dose response curve, right? So, yes, Christina.
How do you know it's just not a behavior?
We don't. We don't. And that's the bugaboo, I would say, with all of these studies because, and I can't tell you how many papers I review where I have to say, you know, that people are imputing all kinds of things to these bacteria. And I'm obviously pro that hypothesis, but we don't know that women are not getting continuously re-exposed to partners who have these bacteria who coincidentally might have the highest viral loads. So confounding by partner's viral load is the absolute black box in these analyses that no one has been able to figure out. I don't know, Scott, do you want to say anything about that? I find it very frustrating because, I mean, we don't know anything about the partners. And if partners were more likely to have these BVAB, the women were more likely to get the BVAB from the partners. The BVAB in the men increased their likelihood of shedding, which they might with urethritis. Then this could all be a reflection of that. So, yes, good observation. Did you have something else in mind with behavior, or was that mostly what you were thinking?
You could just look at number of sex partners. Then, of course, I thought maybe you're smarter if you have orange bacteria.
Yeah. So, Christine, I think that's a topic for my next grant. Actually, that's a great idea. It's like herpes and Alzheimer's disease. I love that idea. So Christina was just bringing up the point that asking, could behavior be impacting these relationships? And that was my response to the first question. I'm sorry I didn't respond to it. I didn't repeat the question. So I really think we don't know. I think it's a really, really critical question. I'm going to show you some partner studies that, that I think help answer some of this, but not completely. And I would say the definitive study has not been done. Partner number is a really imperfect measure, I would say. Yes, they have partner number here, and I think they've looked at the behavioral data, although the behavioral aspects of the study have not been, to my knowledge, published yet. They were obviously very eager to get this out, although I have heard Doug talk about them. The challenge with partner number in these young women is that they tend to be relatively low in these studies. So women increase partner risk in many studies of HIV acquisition in African women. Partner number is very, very difficult to pin down as a risk factor because it's mostly the partner's risk that's informing the risk of transmission. So many have one partner, maybe two partners. Even if you look in our data from the VOICE study, that was the case. Scott, you were going to say something.
Just that in relation to this study in particular, I think it's interesting that what they chose to do, what I think perhaps because the sample size wasn't that big, is that they looked at the association between unprotected sex and HIV. And there was a moderately strong association with a p-value of 0.06, which is in their supplemental tables. And because of that, they said it wasn't significant, so we didn't adjust for unprotected sex. And looking at their p-values, I wonder if they did that, if it would wash away.
Yeah. So Scott was making the point that in the supplemental tables, which I'd forgotten about, there was a marginally non-significant relationship with report of unprotected sex of P06. But, you know, again, the numbers are small. You're dealing with self-report. My guess would be that many of, if not most of these women were having unprotected sex. I don't know what the pregnancy rates were in this study. That would be another interesting biomarker to look at. Certainly Y chromosome would be interesting to look at. So lots of things, I think, to substantiate that. Sylvan, did you have a question? Comment?
Did they look at all its circumcision status?
Question is, did they look at circumcision status in the partners? Not to my knowledge. My guess is that almost all these men would be uncircumcised, but I could be wrong. Okay, so let me just keep going a little bit because I want to make sure we have a little bit of time for more questions, but it's good. So please ask questions in the interim. The other thing that this group did, which I mentioned, was to look at HIV target cells in cytobrushes. I think I said biopsy, but I think they got cytobrushes here. And what they showed, which you can take a look at, is all these, you can see the trends with those increasing diversity of bacteria, but increases essentially in many of the inflammatory mediators that we're talking about as we think about risk of HIV. Now, Scott, presenting your work now, just to sort of say that that study was very, I think, unique and pioneering because it prospectively followed these women with this intent. What many of us have done, or several of us have done, has been to take advantage of saved samples from previous studies and try to look at this as well. And Scott and his group have recently published, and I'm just going to bring these all up, a nice paper, actually presented at IAS, but published in the Lancet HIV, or Lancet ID, right? Just recently, earlier this year now, right? Okay. Looking at a similar question, so samples from women from several sub-Saharan African countries, vaginal samples, used to generate a case control or analysis to sort of say, could we look at women who acquired HIV in these cohorts compared to those who did not, and could we look at differences in the vaginal bacteria? And indeed, this pretty much showed what we just said, that women who acquired HIV in this cohort, there were 57 cases versus 57 match controls, keep that number in mind, were more likely to have a lot of diversity, as shown there. And then some specific bacteria actually were more associated with the risk of getting HIV. And when you look at the adjusted odds ratios for individual bacteria, okay, so just remember, we're now getting down to these individual bacteria. Who do we see? We see our friends Eggerichella and Leptotrichia, and remember, those were the ones in that old analysis of Sujatha's that were most associated with the abnormal AMSL criteria. So again, this gets back, I think, really nicely to Christina's question. Is this because these bacteria are doing something special to the vaginal environment, or perhaps are they a marker of an exposure specific to unprotected sex to male partners type? We also said Megasphaera, Scott did, and Mycoplasma hominis in there. So very associated. We used a very similar approach. Some of you remember the VOICE study, which was a study that I was involved with that looked at oral and vaginal pre-exposure prophylaxis with tenofovir in sub-Saharan African women. We used saved samples from that study to do a very similar analysis. We used quantitative PCR in this study, and we showed essentially a very similar pattern. And again, I won't take you through this, but we showed that, again, Leptotrichia sneathia right here, and Eggerichella species. And the one important point about this study is this is actually about 200 incident cases of HIV. So about three times what Scott was able to look at in his study. So somewhat more, I think, narrow confidence intervals, but really nice to see some confirmation here. The other very interesting thing that we were able to show that I don't believe reached statistical significance in Scott's analysis was that women in the VOICE study who had Lactobacillus crispatus were significantly less likely to get HIV. So again, lots of confirmatory analyses from lots of diverse situations. And when you compare these two studies done in the same lab by Sujatha, David Fredericks et al., this is what you found. So Scott's study in East African women, our study in South African women, that was the VOICE study pretty much, and that's where most of the HIV-infected women came from. Again, we were able to show that Lactobacillus crispatus was associated negatively with HIV risk. And then lots of agreement here. The Megasphaera is a little bit of a weird thing because we couldn't, I believe that you could not in your study did not detect 1 and 2 separately, Scott, but we did. So I would say some pretty nice agreement, and again, consistent with our previous hypotheses and previous observations. Now, what about transmission? And again, I think I've presented these data or showed you these data before, but I do think it's worth reminding people that it's not just acquisition of HIV in women, but there's also a relationship to their likelihood to transmit HIV to men if they are already HIV infected. And these are just some nice data from the partner study that was done here, of course, led by Connie and Jared, looking at women who had HIV and whether or not they had BV before the visit at which their partner's HIV was detected. Okay, so there are discordant couples brought in. The woman has HIV, the man does not. You look at whether the woman has BV or not. You follow the man for HIV acquisition, and you see whether there was a contribution of that pre-existing BV to the likelihood that he got HIV infected. And indeed, Craig Cohen published this, that was the case. And it wasn't just the case like a little bit, but it was the case quite a lot, and it actually increased your risk in terms of the more severe your BV was, right? If you looked at that Nugent score and you had a Nugent score of 10, you were about 7 times more likely to transmit HIV to your male partner. The obvious question here, right, is, well, was BV increasing the woman's genital viral load, right? Because again, that's the main mediator of transmission. And actually, let's see, did I write that? No. And actually, that was the case. There was a slight increase in HIV risk shedding in these women who had BV, but it was only about 1/3 of a log, and it wasn't enough to explain this increased risk. So something else was going on there. Maybe it was transmission of those specific bacteria. This group hasn't looked at those samples to see if that was the case, but you could imagine maybe if the woman had Sneakia, leptotrichia, transmitted that as part of the BV sort of situation, maybe that provoked more target cells in the male urethra, and maybe that was what was mediating the risk. Again, fascinating questions, and we really do not know the answers. Okay, so just to wrap this part up, and then I'll try to finish in about 10 minutes, I think it's pretty clear, hopefully I've made these points, that the older epidemiologic data, relying on gram stain, Nugent score, AMSL criteria, I think definitely support the increased acquisition risk by about 60% in women with BV. In the fresh cohort, the high diversity communities increase HIV risk probably about 4-fold. So pretty striking and quite a bit larger than the old meta-analysis increase, probably because you were looking at young women with very high HIV risk in general. There were specific bacterial taxa that have been associated with reduced or increased risk, and that's what Scott showed and what we have showed subsequently in the voice data. And we know that BV increases risk of HIV transmission to male partners from Craig Cohen's data. So then the question is, how does this happen? And I'll just sort of leave you with a few intriguing, I think, hypotheses. There are lots of thoughts about how this might happen. I mentioned HIV shedding, but that's really been inconsistent. Sometimes we've been able to find that with BV, sometimes we really haven't. And as I mentioned in the Partners in Prevention study, that really didn't explain the increased risk of HIV transmission to these women. So we really don't know what it's about. I do think that the lactic acid production by Crispatus and Jensenii is a really critical thing. I realize that I need to update this because I should have one of the women from Black Panther. I think it would be much more compelling. But Wonder Woman's pretty good. So what is it? I mean, really, we're talking now about probiotics, right? There's a study that Craig's leading with a lactobacillus probiotic called Lactin-V. And he's using it for recurrent BV. I'm hoping the results will be out later this year. But what is it about these bacteria that is clearly so good? We used to think that they had to be hydrogen peroxide producing to actually affect this protection. I don't think that we believe that anymore. It's really been debunked in the laboratory and in other settings. But Gilda Tachadian from Melbourne has done some really nice work in vitro looking at lactic acid production by probiotic lactobacillus species. And I think we're really getting the sense that that lactic acid is probably quite important. We're now moving into a field or time where we're using more functional assays and omics. So we're using proteomics, which tells you what proteins are expressed by whoever is there. We're using metabolomics, which I mentioned before, are a signal of metabolic activity. And when you start to look at that, I think you get a sense that the lactobacilli are probably producing things that are more than just the lactic acid. And I won't take you through these in the interest of time, but I will show you a couple of really interesting, I think, more recent proteomic and microbiomic sort of data. So again, proteomics is a very intriguing field. It's the kind of thing that if you don't know what you're doing, you can make a lot of mistakes. So you really need to sort of read the literature carefully and work with somebody who understands how to read the data. But really, it's giving you a window, not so much who's there, but what genes are activated and what proteins they're making. Okay. So this is a very nice study looking at asymptomatic women in Africa. And looking at the genital inflammation as measured by all of those cytokines here. This is a heat map that just shows you what happens as you go from that optimal vaginal flora down the left there, as we'd seen before, so the Nugent score being totally normal, and then as you go up here. And you basically see this increase in these inflammatory markers, which we've already talked about. And the nice thing about the study is that they included women who had herpes, trich, gonorrhea, and chlamydia. A lot of these studies either don't really talk about that or they don't bother to look at it. What I think the point of this is to show you is that if you look at who's inflamed down here by these cytokine sort of panels, they are mostly women who have disrupted vaginal bacteria as opposed to the traditional STIs. So that's a main message. So again, coming back around, right? Why is VV itself a risk factor for HIV? Because you do have a disrupted inflammatory profile, and I think that's really important. Another nice paper from Vicky Jesper's group and Janneke van der Weigert, who have a nice cohort in Rwanda. I believe this is from the Rwanda cohort. Looked not only at the diversity in these women, but then again at the proteome. And so if you're interested in this, these are some of the things that you can associate with the presence of these good species. And what are these? They are proteins that are associated with good mucus production, a good cytoskeleton, and very interestingly, epithelial cell junctions. So one of the things that we think is that when you don't have these lactobacilli, you may not only not have the lactic acid, but you may actually have disruption of the epithelial cell barriers that promotes biofilm formation that's associated with BV, which is a very bad thing, but also may create portals of entry for HIV and for infected cells and inflammatory cells. So again, we're kind of trying to get down to a more functional picture, I think, of what's going on here. And it's complicated, but hopefully we're getting there. The last thing I'll just mention is this whole question of whether antiretroviral therapy and tenofovir-based PrEP are affected by the vaginal microbiome. Probably you've heard a lot about this. And let me just come back to that and then we'll wrap this up. So I mentioned before that PrEP is really doing great in men who have sex with men, certainly in the United States and in many westernized countries. Story with women is not so good. And the question has been why? Is it because they don't take it? So is it all because it's non-adherence? They don't really think it works? Is it because tenofovir doesn't get into the cervical tissue the way that it does into the rectal tissue? That's definitely true, unfortunately. One more biological sexism example. Is it because vaginal bacteria create an environment that is hostile to tenofovir? That's another possibility. Or is it because of inflammation, right? So there's something about inflammation that makes it, increases the bar for protection at the cervix. Okay, so I think all of these things deserve, again, more time than I'm going to have to do today. But I will just point out a couple of things. If you look at the CAPRISA data, and CAPRISA was a study of pericoital, so pre and post vaginal intercourse tenofovir gel, reduced the risk of HIV acquisition by about 37–40%. You go back and you look at protection afforded by that gel, tenofovir gel, what you find when you break it down by the presence of lactobacillus in the vagina is that there's a difference. So study that was published recently by Eclat and colleagues and also Lyle McKinnon shows you that the probability of HIV infection in the CAPRISA cohort over time by women who are lactobacillus dominant, they don't separate out Einers and crispatus here, although they do in the supplementary results, showed a pretty big difference. In other words, you could see that in the placebo versus the tenofovir, there was a very nice separation, and that was what you saw in the original study. When you take women who had a non-lactobacillus dominant, meaning that they were in those community state types 3 and 4, the gel didn't work. And that was a really big surprise to many people. So what was going on there? Well, there are probably a couple of things. The group just published this year in Science Translational Medicine, I think, a really interesting analysis looking at this by inflammation as measured by cytokines. And what they did was to measure a bunch of pro-inflammatory cytokines and say whether women fulfilled this criteria for inflammation or not. And what you're seeing here is that the efficacy of tenofovir gel ranged from 38 to 57%, so quite good. Reduced your risk of HIV acquisition by 1/3 to over 1/2 in women with lower inflammatory cytokines. So those are the women in blue who experienced protection, and that's tenofovir efficacy on the bottom. But in the women who were inflamed by measure of these cytokines, the gel essentially didn't work at all. Period. It was 3 to minus 147%. And the most amazing thing to me is that you can go from relatively low cytokines up to more, and you see again this biological trend or this quantitative trend in the other direction. Okay. And furthermore, this was seen even when women used the gel. So they actually had pretty good measures of adherence here. Even in the women who used the gel every time they had sex, there was a really marked difference in the efficacy. So what on earth was going on here? So again, this is just, and Christina, to get to your point about trying to separate this out, because again, getting back to the big question, maybe women who use the gel more were having higher exposure. Obviously, they're having more sex, right? But maybe they were having sex with higher risk partners who had higher viral loads or higher likelihood of being colonized with the BVAB. And that would create inflammation. So again, we still don't know that this is not just a gigantic story we're telling ourselves. And that's the beautiful thing about science, right? We really tell ourselves stories and then we try to disprove them. We haven't been able to disprove this yet, I think. So the higher number of sex acts was significant, and this is just a table where they looked at everything. And herpes was also significant, even when you didn't see inflammation, which I think is actually fascinating. And it is difficult, I think, to disaggregate sex and the gel use and all this stuff. But nonetheless, there's a really strong signal here, and what does that mean? Eclat published an analysis in Science, again last year, that purported to show that tenofovir was essentially being eaten by Gardnerella vaginalis. She did this work in vitro. It hasn't been shown to be the case in actual women in vaginal fluid, although Betsy Harold has some very interesting data to support this hypothesis. So I do think that there was something going on here with topical tenofovir that is modified by vaginal bacteria. So it's kind of like a double whammy. Not only do you have the suboptimal environment, but you're even less likely to be protected if you use topical product. This is not the case for oral PrEP. The Partners in Prevention study, Renee Heffron, published a very nice paper, actually 1.5 years ago now, showing that in the Partners in Prevention study, the effect of protection from oral tenofovir products did not matter by whether you had BV or not. So clearly, oral tenofovir gets where it needs to go, and it's not affected by this. Okay? So what are we going to do next? So that's sort of the whole situation. It's not particularly good for women, except to say that oral PrEP, oral tenofovir, even though it doesn't get to the cervix as quickly or as easily as it does to the rectum, if you take it every day, you probably are going to get pretty well protected regardless of what's going on with your vaginal bacteria. But there are barriers. I'd like to show this slide because it's from 1919, and women were just as desperate then to spend a lot of money on worthless products as they are now to protect themselves from or to treat their symptomatic vaginal complaints. Some of you have seen this. This is a slide that a friend of mine found an old Cosmopolitan in a garage sale. And the good thing about doing vaginal research is that you get all these crazy things. And so actually she took a picture of this for me and I made a slide. This is a question from Cosmo, January 1949, why is her husband so cruelly indifferent? And the bottom line is that she has bad vaginal odor. So I'll just cut to the chase. But the reason that it's an ad is it's an ad for Lysol. And if you haven't seen this, there are lots of Lysol ads for douches, actually, from that time period that we're talking about in 1949. And even doctors recommended to ensure daintiness alone. So I point that out, like it's sort of funny, but it's also sort of tragic because we are still in an era where vaginal products are totally unregulated. I mean, you can get pretty much anything to put in your vagina. They're treated as cosmetics, right? So the same care that goes into whatever you put on your face is what you can put in your cervix, essentially. And we really have had, I would say, despite the fact that we know that this disrupted environment is clearly a risk, we really don't have very many ways to fix it. Here are some of the things that I like to think about. I mentioned lactobacillus repletion, which is under study. Craig Cohen running that study, which I hope we'll find soon. Biofilm disruption, we have a paper coming out in CID describing the results of something called Toll 463. Julie's ran the study here with us in Birmingham. That is a biofilm disruptor boric acid, EDTA product that actually did work to treat BV and VVC or vulvovaginal candidiasis. And so I'm hopeful that that might be something we might look at. Vaginal hygiene interventions have tried very hard to work on that. New antimicrobials, not so many, I would say, in this field. Menstrual suppression, an interesting idea, and we'll talk about that in a second. And again, everybody's pointed out creative approaches to partner management. We really need to figure out what the male factor is, I think, here. Female factor, certainly we know that women transmit BV to each other. For the HIV epidemic, I think that's not quite so relevant. And then modifying the hormonal environment. And that's the last thing I want to leave you with, because I think this has some interesting potential. And this paper has just been published in PLOS One, I think yesterday. Many of you know about the NuvaRing, which is the contraceptive vaginal ring that has estrogen and progesterone in it. And remember, for contraception, you really just need the progesterone. The estrogen is really just there to regulate the menstrual cycle. Okay, so with progesterone, you get unpredictable bleeding. Giving women progesterone, one of the reasons women stop progesterone-based contraception is unpredictable bleeding. So it's a very interesting and nice way to afford contraception. There have been some data to say that estrogen delivery, for reasons that are pretty sensible, namely it supports the epithelium, probably is good for the vaginal environment. And I'll just point out that this project, which has just been published, looked at the impact of using this ring in Rwanda women, and what they showed was that whether you used it continuously, meaning you didn't menstruate, or whether you used it cyclically, so you took it out for a week so women would menstruate, you had a really profound effect on the vaginal microbiota. So wouldn't it be interesting to have a ring that maybe gave you protection from unprotected pregnancy and also optimized your vaginal environment? And I'm very hopeful that there may be something here. And we are actually studying this. We've got a study that just finished in Kenya, where we've enrolled 120 women and randomized them to cyclic versus continuous. So very similar to the Rwanda study. But a lot more measures and about the same number of women. And we actually have completed this study and hope to present data later this year. And we have a smaller study here in Seattle that has some preliminary data that's very encouraging, but I don't want to show you yet because I don't want to jinx it. So I think it's really interesting. I mean, maybe this is one way we can sort of try to optimize it. But again, it doesn't involve partners. And it is only one way that I think we need to move forward. And I think part of the challenge is that, as you saw from my very first slide, that this is an area that really requires incredible interdisciplinary involvement, right? You have to have people who care about women's reproductive health. You have to have people who care about a vagina and aren't just interested in the virus, which is a challenge. You have to have people who understand anaerobic microbiology and understand the complexity of immune inflammation. So it's really an incredibly meaty area. But it does take somebody who, it takes a team of people who really, really want to, I think, collaborate and communicate on some of this stuff. So I'll just stop by thanking incredible team. This is all Seattle-based people who have been involved in these work, and I think you recognize many of these people. Jen Bulk is actually, I want to thank specifically for some of these slides, and also Scott, who's not on here but should be. And also, this is a FICA research team where the FICA project is going on. And I'll stop there with some Alabama pictures. So thank you very much. I think I have 1 minute. I can hang around a little bit, but then I have to go to the principal's course. Thank you very much. Sorry, I hope it wasn't too complicated. Thanks.